Yes, good evening to all the participants. So, welcome to the day 10 session on molecular talking analysis as well as molecular dynamics simulation. Before proceeding on to this topic, can we recap the last session topics? So, what are the last session topics? What exactly did we discuss? Even the SVC students can answer. See, nowadays SVC students are not answering. Either unmute or you can message in the chat box. Okay, utilize this opportunity. I think you will be getting passed out, isn't it? Within a semester after completing project. Interact so that you can also assess yourself. Good. So I got information from a participant. Molecular talking. We discussed on what is molecular talking and what is molecular recognition. Like in biotechnology, we come across different areas of biotechnology like immunology, enzymology, signal transduction, and ion transport. So like this, we have so many kind of types of biology. We go and dig in deep of types of biology. Whatever we study the molecular recognition in these types of biology can be even microbiology, molecular biology. So this we try to use it in computational part where checking the binding affinity of ligand with the receptor in computational process called molecular docking. Once again, I'm telling molecular docking is a process to check the conformer of a ligand receptor by complex through estimating the binding energy. If so, we have also discussed something about the importance of energy minimization, which is a part of the protocol of molecular docking. So, if you want to do the molecular docking, we have to do a part of the step to make the molecules more stable. And we discussed on what are conformers. In the definition itself, we can see the conformers will come. Okay, so the ligand dock with receptor in multiple conformers. It means that the ligand as well as the receptor are in flexible state. If the ligand is flexible, the ligand can bind with n number of conformers, the target protein. So that's what we call it as conformers. Conformers also can called as poses. Okay, that is a different. That's a terminology for the conformers. Different poses of a ligand binding with the receptor. Docking classification. We have seen extensively on docking classification. Like we have discussed, like small molecule receptor protein docking and nucleic acid protein docking. We have seen the macromolecule docking, that is antigen antibody docking, protein protein docking. So like this, we have seen the types of docking. Other than this, we also seen the docking based on flexibility and rigidity of ligand and receptor. Okay, so out of all these four types of docking based on the rigidity and flexibility, the one thing which is not possible is rigid ligand and flexible protein. Okay, so that is actually not possible. We have discussed, and then we have discussed something on shape complementarity, chemical complementarity. So, what do you mean by shape complementarity? I told you there's most of the shape complementarity we use in pharmacophore based methods. So, pharmacophore based modeling mapping, this comes under a prior step to the molecular docking in sequence of stages of drug design. So where we generally check how effectively a ligand is bound with the receptor. Only the fitting alone we do try. Whatever fitting we try, we calculate this fitness through fitness score. Okay, and this is what we're reporting in pharmacophore based. That is a hybrid approach of pharmacophore based modeling. If that is, uh, I mean, if pharmacophore is restricted up to the fitness values, the molecular docking can estimate as the binding energy based on chemical complementarity. So I believe one of the participant who asked the question is not available. When she is present, I can also answer for her questions. I told you earlier that if you have any doubts, related to any of the sessions, you can also ask me in private WhatsApp. Okay, she has communicated me, but presently she is not available. Even if you are interested and if you don't, if you have some kind of doubts, if you can't able to ask in this session, you can ask me like that. Okay, in the WhatsApp, so that I can recollect and mention it. Okay, and we also discussed something on the talking scores, the scoring, uh, the grid calculation. Talking score is most important, and even search algorithm in talking. This is also important. Actually, if you see in talking, there are two most important things we have to remember. One is what algorithm we are using for talking, and the number two is, so what is the scoring function we are using in talking. Okay, so these two are most important essentials which we distinguish the freewares from the commercial softwares. We also seen plenty of freeware tools and the commercial tools. For freeware tools in Autodoc, if you see, we have Autodoc 4.0, Covalent Autodoc, and if we want to go with dock in multiple kind of elegance with the receptor, we have Autodoc PYRX, and what else? Autodoc Vina. Okay, so these are some of the Autotalk tools. And yesterday, whatever we practiced, that is Autotalk 4.0. And we have seen sequence of steps and even the steps what we followed in the protocol. I just also uploaded in the WhatsApp group. Okay. So other than this, did we discuss something else? Other than this, did we discuss something else? Any other participant like to mention if I missed anything? How many steps are there in? I showed you on yesterday in molecular docking using Autodoc 4.0. How many major steps? I've sent you the protocol. How many major steps in docking completed on yesterday? This is my question. To see the protocol, first you just download and check the protocol. How many major steps I have completed? Because I'm going to continue that today. I have to illustrate on the docking analysis. Yes, very good. Six steps. Okay. Six steps we have discussed in molecular docking. Can we see this protocol? The protocol, what we followed, actually, I sent you this Autodoc tutorial. If you remember, I can also ask you one more question. So, six steps is there, isn't it? So, other than six steps, uh, so this is the protocol. Yeah. How? What are the tools? What are the software tools we have to install to work with Autodoc 4.0? The tools or softwares used in Autodoc 4.0. I also mentioned about this. Very good. Python molecular viewer, EDU Python molecular viewer. One tool you mentioned. How about the other three tools? Yes, participants only. The interacting participants are interacting. How about the others? Come with your answers, right or wrong. Nothing. Good. Rasmol, but tried well. But I didn't show you on Rasmol. So EDU PyMol, MGL tools is okay. Other than this, Raccoon really, I didn't explain Raccoon. For the Autodoc, can I show you once again so that you won't forget? Okay, so I'll be showing you once again. So the tools what I showed you, even I mentioned about the bit version, 64-bit version, 32-bit version. If you have that Autodoc tool, okay. If not, you have to remember. So can you able to see this? Now you can type the answers in the chat box. I have showed you the tools. What else is missed? I believe I showed in large icons. So what else I missed? You type and show. Chimera, very good. Chimera. Other than chimera, Autodoc. Okay, we have Autodoc suit. Okay, so these are the four tools. Again, I'm telling you, the tools we have used is EduPymol, MGL tools, Chimera, and Autodoc suit. These are the four softwares we have to install into four different websites. I mean, we have to download from four different websites and we have to install to work with Autodoc 4.0. And then we've practiced the 6 steps. So before proceeding the step one, I mentioned you to first uh, mention the path of Autodoc. Okay. Then I told you after fixing the path, then only we can go for docking. So before starting, I mean, starting the first step of the protocol, I told you to have files. What are the files we should have in one folder? So the Autodoc 4, AutoGrid 4. This Autodoc 4 and AutoGrid 4 are exe files, executable files. So if you install these four softwares, you also can get these two files in the program files folder. Okay, in the MGL tools, you have to take and paste it in one folder. After that, even you can keep a tutorial, like whenever you want to work, you can use this, you can open the tutorial and work with it. And the next two important files to start the process is a ligand, okay, a ligand molecule and a protein molecule. So the protein molecule initially I had is this protease, and the ligand molecule I downloaded from the NCBA PubChem. If you want to download the protein molecule, use PDB database. Okay, so using the PDB database, get the 3D structure. Similarly, in any drug, whatever you're going to download, take only 3D structure. Don't use 2D structure for docking. That is not appropriate. If you don't have a 3D structure, convert that structure as 2D, I mean 3D structure of a ligand. Then only you can go for docking. Okay, you can't able to use 1D format like a smiles canonical smile format of a ligand. You can't even use the 2D format of what only the 3D format. If you have, you can use it for docking. Okay, so these are the 5 files initially had. But after docking, after completing 6 steps, you can find so many intermediate files are formed. Initially, we tried with 5 files, but now we have around many files. Okay, now if I want to analyze the result, okay, remember the last file what I saved is the BB underscore protease underscore complex. Okay, that is BB stands for ligand, wherein on B, protease is the protease protein, that is a target protein. So now to analyze it, I suggest you to use Discovery Studio. Even though I've described like how to analyze using the Python molecular viewer, okay, but that is something complex and it's not user friendly. So the one whatever I'm going to teach you, other than whatever protocol I've sent, is to analyze the results using Discovery Studio Visualizer. See the protein, the protease protein, whatever I dropped, okay, you can able to see the secondary structure in solid ribbon format. If you have this kind of this blue color kind of thing, this how it looks like. This looks like strands. So these are beta strands. You can able to see the beta strand in the protease protein, or which is connected with loops, and this is our ligand, which is in stick model. Okay, now if you want to see the 2D interactions, okay, so here in view interactions you can find show 2D diagram. In view interactions you can see show 2D diagram. Now I'm going to click this. If I click this, you can able to see this 2D diagram. So this is the 2D diagram. So this is your ligand, okay, talking with the amino acids of a target protein. And you can see the types of interactions. Basically, we have covalent interactions and non-covalent interactions. So one of the participants asked me on yesterday night at 8:14 p.m. that, sir, still I am unclear about the ions. What are the roles of ions, okay, in molecular docking as well as MD simulation? Okay, she asked me to explain on today. So just because I'm going to provide the recorded version, I'm going to explain now. So actually, if you see the ions, okay, so here if you have O stands for negative ion. Okay, so if it is an anion, and if H plus from isoleucine amino acid is going to contact O, that is positive ion cation is interacting with the anion. If you have these kind of an positive and negative ion interactions, these ionic interactions are strong. These ionic interactions are strong when compared to non-ionic interactions. Okay, so how the binding energy is estimated, I told you, based on number of hydrogen bonds formed. So even if you see the hydrogen bonds, you can able to see the types of hydrogen bonds like conventional hydrogen bond, carbon hydrogen bond, and we also see pi donor hydrogen bond. So like this, we have types of hydrogen bonds. Of course, hydrogen bonds can also give a strong bonding interaction between a ligand and the receptor. Okay, so now we can see these three types of hydrogen bonds are formed. So obviously, you can find this, you can able to conclude this compound is binding effectively with the target protein. Okay, so three hydrogen bonds, and I mean, nearly four hydrogen bonds are formed. The green color you can see like that. And other than this, we also find van der Waals, pi cation, pi sigma, pi alkyl bonds, etc. And if you want to get a good kind of a display, okay, so if you see this ligand, the ligand is present in the form of the kind of line model. If I want a kind of stick model, so I click apply. Now the ligand is found in stick model. The same thing I'll be also showing it in 3D. Now, whatever you find in a stick model in ligand, if I want to see this residue, okay, in disk model, so see the lightly you find the borders inside. Like this, I can find. And one of the thing most important is if you want to check the bond length, click this show distance and click apply. If you click apply, obviously get the bond lengths. But here, before showing the bond lengths, I can show you one more thing. So if you see the favorable bonds, we have bonds like hydrogen bonds, electrostatic bonds, hydrophobic bonds, halogen bonds, miscellaneous bonds. See, this is what the participant has asked me. Other than these ions, positive ion and negative ion, I already told you this also gives a good affinity. I mean, this can show you a good kind of an interactions that are positive and negative. Other than that, even some of the ions like chlorine, bromine, iodine, okay, and sulfur, this can also be like this can also work as a cofactor for the proteins. For making a proteins more stable, we use these kind of a metal ions as a cofactors. Like how enzymes can used as a cofactor, similarly the metal ions in the protein can also stabilize the protein structure. Okay, so we should not delete these ions if we have these ions in the protein. You remember I told you in protein structure coordinates we have atoms as well as heteroatoms. The atom coordinates are amino acids of the protein. HET-ATM coordinates are the water molecules. Other than water molecules, we can also find chlorine, bromine, iodine, sulfur. Okay, if you have some other metal binding proteins, okay, so we can also ion binding protein like hemoglobin. In hemoglobin, iron is the metal. So whatever iron we find in the form of porphyrin structure in the hemoglobin, that gives a stable structure for the hemoglobin protein. So if you want to, if you use that as a target, you can't delete the iron molecule. Similarly, for chlorine binding proteins, you can't delete the chlorine. So you can't able to delete the chlorine. So like this, we should retain the ions. So ions play in multiple roles. One is in the interaction, cation and anion interaction. The other is ion interactions. I mean, ion channels. Some drugs are transported only through the ion channels. I told you an example is sodium and potassium ion channels. This transport can also give some kind of a knowledge on how the drugs are also transported inside the desired tissue. That is one of the mechanisms. And the third is even these ions are present along with these proteins. These ions should not be deleted. during the docking, but the water heteroatom molecules like water molecules that can be deleted. So if we delete that water molecules, then only it can able to know whether our ligand is binding with that appropriate hydrophilic binding sites or not. How we check the hydrophilic binding sites? If and if it deletes the water molecules, and if our ligand is going to bind in that hydrophilic binding site, it can determine. But if water molecules are present, it hinders our kind of an interaction. So you got it. In heteroatoms, we have to retain the ions, cofactors. We can't delete. Similarly, even in empty simulation. In empty simulation, these ions presence in the proteins can give complications. If you have the ions, we have to include some code in the simulation, MD simulation. So when I explain about the MD simulation, we can see about that. Okay. Of course, for MD simulation, one should have a knowledge of Python programming, Perl programming. You believe it or not? Okay. So simulation is something complex. Okay. So even many of the PhD research scholars in IIT Madras take work on MD simulation in PhD research. So that much information is there. If you work on complex proteins, okay, if you work on multiple proteins, okay. So, but I will be giving you some basic outline on simulation, and even this laptop, even i7 and i9 laptops are not sufficient enough to explain on MD simulation. So we should need a kind of a workstation or a server. If only if you have a good configuration of a server or a configure or a workstation, then only we can able to work with simulation. And even showing demo on MD simulation can take for more than 2 days. Okay, 2 days continuously, 48 hours. So if you want explicit training on MD simulation, they have 15 days training. Not myself, and another kind of a faculty can train you. If you want to get trained in MD simulation specifically, you can keep in touch with me in WhatsApp number. I can provide you the contact. There is much more to learn. They are asking to come to directly to college to learn, to acquire the skills. 15 days offline training in MD simulation. Okay, now let me come across the different kind of results. I told you, if you want to see only the hydrogen bonds, you deselect the electrostatic, deselect the hydrophobic. So even this deselecting, selecting is possible. Okay, if you don't want a pi alkyl, okay, I just remove the pi alkyl, apply, pi alkyl is removed. If you want to retain only the hydrogen bond, you can retain the hydrogen bond. Even if you want to delete pi cation, you can deselect it and you can apply it. And if you want to check the distance of the hydrogen bond, click apply. Now you can able to find the hydrogen bond length. So whatever show distance can show you the hydrogen bond length. So till now, whatever we have discussed is how to see the 2D interaction of the protein receptor complex. How about the 3D interaction? So till now, we have discussed on 2D interaction. Now we can see the 3D interaction. To work on 3D interaction, okay, one can go to this uh, structure. Before going to the structure, go to the view hierarchy. If I click this view hierarchy, you can able to see a chain, and then in this you can see this chain. In the chain, it is in small font only, so only I'm explaining. So a chain is full of amino acids of a protein, but we also have something called chain, and under this chain we have UNK0. This stands for a ligand. So whatever ligand we have selected, okay, so we have to go to the scripts selection. Either you can select ligand directly like this, or you can select you can use the script selection select ligands. So now the ligand is already selected, so it is in yellow color. Now next to that is select atoms within 4 angstrom. Only 4 steps you have to remember. The first step is select ligands. The second step is select atoms within 4 angstrom. Okay. The next step is scripts, ligand interactions, show ligand binding site atoms. So if you do this, you can able to find like this. And if you want to check what are the amino acids contacting with the atoms of the, what are the atoms of the ligand contacting with the atoms of the amino acid of a protein, that we can't able to see in 2D interaction. In 2D interaction, we can able to see only what amino acid and amino acid position interacting with the ligand. But here you can able to see UNK0 in the hierarchy part. You can able to see UNK0, 05. Okay, so this is actually that is the ligand. O5 position of the ligand is interacting with HN position of the isoleucine 3. I can tell you once again, O5 of ligand interacting with HN of isoleucine 3. So whatever you have seen, isoleucine 3, where is the isoleucine 3? So we can't able to find the atoms here. But in this hydrogen bond monitor, we can also see what atom of isoleucine. O5 with HN. Okay. So this is how whatever we talk, okay, if you want to take an additional parameters like the atom positions, this can also be included in a table. So if you see that molecular docking, the most we have three important results. The first important result is the 2D interaction. The second important result is the binding energy, whatever we estimated on yesterday. You remember I showed you nearly minus 7.6 something calorie per mole. That is one of the data. And the second data is the atom position of the ligand and the atom position of the amino acid. So that we can able to find it from here in this hierarchy. Okay. So, and after doing these three steps, select ligands, select atoms within 4 angstrom and ligand interaction, show ligand binding sites. And the third important thing is to get a 3D interaction image. To get the 3D interaction image from the window. So whatever I have, so I have only a part of the binding sites. If I want all the binding sites, I can select the entire protein. I can select the entire protein on whatever amino acid or the whatever amino acids you want to select, you can select that and you can convert it into different models. For example, isoleucine 3 is one of the binding site. I have selected this isoleucine. Now right click, display style. I'm going to use this isoleucine as ball and stick. And then I go to the graphics. Instead of background color black, I click like white because in the journal publication, we are going to use this, isn't it? If the 3D interaction, see, this is my binding site amino acid. And you can also see this green bond, this green hydrogen bond. And able to see a green hydrogen bond is there. So green hydrogen bond is sharing with the ligand. This is an amino acid. So like whatever amino acid we have selected, if you want to label it, you can use structure label side. And then after the labels amino acid chain, you can keep something like an amino acid. In amino acid, you can use something like the 3-letter ID and insertion code. If I click apply, you can see this isoleucine 3, but it's in very small font. If you want to increase the label with a large font, I'm going to increase the label with the large font. Now we can able to see the isoleucine 3, the isoleucine 3 is interacting with that particular atom. So like this, you can take a 3D image from this. So this is a third output. Okay, so this is how we can analyze the docking results. Now let me take you to the MD simulation. Till now, whatever we have discussed, how to analyze the results of molecular docking from Autodock to in Discovery Studio Visualizer. Okay, so to check the kind of a results, okay, other than the Autodock, whatever we calculate in terms of binding energy, we can also add the have bond interactions. See, this is what bond interactions, atoms from the compounds and atom from the protein target. So this is one such data. Okay, these are two data, and similarly I have shown from another paper. And also I showed you, I mentioned you either it can show either 2D interaction image or 3D interaction image. Even in ovarian cancer article, whatever I have provided, there I have given this docking result in the form of a graph instead of table. Okay, so in now let us come to the MD simulation. In MD simulation, okay, we also find similar kind of a molecular modeling concepts. So we study from classical mechanics to statistical mechanics, and then we study up to the quantum mechanics up to the macroscopic level. If you come across MD simulation, I told you the difference between MD simulation and docking. You remember, in docking, we generally check the affinity of ligand with receptor. If it is over a period of time, we call it as MD simulation. Okay, so in terms of ligand receptor MD simulation. But MD simulation can also be used for other purposes. For example, if we want to get a 3D structure of a protein, I told you, we can use X-ray crystallography. The X-ray crystallography is one of the wet lab technique. So whatever X-ray coordinates we get in terms of black and white color kind of a graph, like even if you take an X-ray, we get white bones in the black background, isn't it? Similarly, the protein amino acids are shown in white color spots in the black background. Whatever we get as X-ray coordinates, if you want to convert it as PDB coordinates, still we can use MD simulation. MD simulation can also be used to check the protein, okay, the effect of solvents, different solvents in protein. For example, if you want to prepare a buffer to maintain a protein for a long storage, obviously, if you want to try whatever you're going to try with the wet lab, we can try the same thing even in computational part. Like this, we can use MD simulation for multiple purposes. So whatever we can work in a wet lab, if you want to do the kind of a simulation in computer, we do that. Okay, so for this, we have, you're using a plenty of tools like abalone, amber, charm, testmon, chromax. Okay, I'll be explaining much on the chromax because I worked on chromax, and even recent publications I'm also publishing on Scroidinger kind of an AMD simulation package. Okay, and Gromos, NAMD is a commercial software. The Gromax is a freeware. If you want to work with the freeware, I suggest you to use the Gromax. Groningen Machine for Chemical Simulations, that is the expansion. And Q and answer are the commercial tools like NAMD. NAMD stands for Nanoscale Molecular Dynamics. For high impact factor journals, publications, we can also try with the Chromax. Okay. Now let me take you to this MD simulation by Chromax. But for the disadvantage in using Chromax is we should have a high configuration GPU. Okay, high configuration GPU, that is, we should have a workstation or a server. We can't able to work in our laptops. MD simulation in Chromax. In my ENRF Tare funded project, uh, I worked a part of a project in IIT Madras, a part of a project in computational part, that is on MD simulation. So in my research budget, I procured a kind of a server in the IIT Madras, and we are working on MD simulation there. I've installed the Chromax and I started working. I and also completed that funded project in IIT Madras. Okay, so one should have such a kind of a good configuration system to work. That's what I want to mean. We can't able to work in even local desktops present in our labs. We should have high configuration server. And if you want to analyze the final outputs of the MD simulation, we have to install other softwares other than Chromax. The tools like XMGR Grace Graphs. Okay, so these are the tools we use it finally to analyze the results of MD simulation. Now let me take you to the MD simulation. In MD simulation, if you see, these are the tools, these are the programs we use in MD simulation. For using these tools, one should uh, use the Linux operating system. So the Chromax, I believe up to 2021, we can able to work only with the Linux operating system. We don't have any kind of Chromax tool for Windows. So the Chromax for Windows is recently developed, but whatever we have, even that is not exactly Windows, we can have a interface. We can have an interface, that is Linux interface software we can install in Windows, and in Windows itself we can work with Linux commands, like how we can use TYGWin. You know this, you heard about this. TYGWin is one of the interface, is one of the tool we can download and install in Windows, and we can work with Unix commands. But if you want to work with Chromax, if you want to install Chromax in Windows itself, we can use that Linux interface. Okay, like PowerShell. Okay, PowerShell is one of the interface where we can able to practice the Linux commands. So if you want to work with Chromax, you have to use only the commands. The command you have to execute only through commands. So we are using whatever programs I showed. For example, if I want to work on solvation, I have to use this as a command: gmx solvate spacebar attributes and the file names. And similarly, whatever programs I showed, these are going to work as a commands. Okay, so what are the different processes we can use with this? If you see, GMX setup is used for to produce a topology, and GMX solvent is for solvating a system. Like defaultly, the protein is kept in water for solvation. But if you're using your protein in different solvent, for example, if it's subject to methanol, what is going to happen with the stability of a protein? So if you're changing the solvent, checking the stability, we can try with this GMX solvate. GMX solvate command, and then we can try with the appropriate solvent, and we can give the file name of the protein, etc. And similarly, energy minimization, you can use GMX-AM, and you can see the similarly. So these are some of the protocols we follow in AMD simulation using Chromax. So before showing you the output, I can mention you about the homepage of the Chromax, and then I can tell you some applications of Chromax. So if you want to know about the Chromax, simply go to the chromax.org. So inside the chromax.org, you have an option like click downloads. Actually, myself, first time I tried in IIT Madras, so it's easier only. If you want to download, install, and work with the Chromax, it takes some time. But to reduce the time, I suggest you to use YouTube videos. We have several recorded YouTube videos to work with Chromax. If you follow the protocol first, initially with the sample file, whatever they have provided, like however I provided demo on Autodoc. Similarly, follow these kind of steps in a YouTube video, you will be getting the result for sure. But for working with your sample proteins, you need some more knowledge. For acquiring this additional knowledge, I mentioned you that if you can contact me, I can direct it to one institute, a popular college where they can train you 15 days offline. That means 7 into 15. Okay, that much hours they can train you in MD simulation alone. Okay, a lot to learn there also. So okay, so to install the latest version, you can use this 2025 like this. And one thing we have to remember, for to know more about how to use etc., one should can go through the documentation. So that just go through this documentation and get some sufficient knowledge. And one more thing you have to remember in Chromax is Chromax tutorial. There is something called Chromax tutorial, which we can able to see in mdtutorials.com. This is another website URL, the most important URL to work with the sample files. I told you we have YouTube videos to work with the sample files. So how, but where to get the sample files and where to get a kind of an API documentation for usage of this protocol. So related to this, you can able to find in amdtutorials.com/gmx. So this is the URL one has to go for. I think I've typed in that WhatsApp. If not, you can follow this. Okay. Now let me explain about this. So if you see this tutorial one is about lysozyme in water. If you put this lysozyme protein, how about the stability over a period of time? Or after 100 nanoseconds, after 200 nanoseconds, we can check, we can fix one particular time, and for a period of time, we can check the stability of the protein in solvent like water. The solvent is the water is the default solvent kept in MD simulation, and even in docking to check the binding sites. But if you want to change the solvent, you can able to change it only in MD simulation. So you can change methanol, you can change some other solvents also. Okay, and related to this, okay, we can also be able to check how about This is DPPC is a kind of a bilipid membrane. If you want to check an effect of a solvent in this kind of lipid membranes, we can also try that. And even if you go with a drug design, if you design a drug and if your drug has to target a bilipid membrane, how it's going to target. So like this, you can integrate, okay, I mean, you can use these principles to your even for MD simulation using Chromax. The third example is protein ligand complex. This is what I have done, okay, and have more than 8 to 9 literature paper, I mean, 8 to 9 research paper data yet to be published. Okay, so soon I'll be publishing this kind of a data with the MD simulation, but presently I believe even I have 4 to 5 articles with MD simulation. Okay, so also go through these articles, whatever, just the important articles I've uploaded in the WhatsApp. Okay, so after doing molecular docking using Autodock, the final ligand protein complex, you take it as an input for MD simulation. The input for MD simulation is the output of molecular docking. Okay, so you use this protocol tutorial 3 to work on MD simulation. This is what most people use in drug design. The remaining thing or the other applications of MD simulation. Okay, these are other applications. This can be used in chemistry. The chemistry persons can use this, and even the polymer technology people can also use the other kind of examples. Now, let me take you to the protein ligand complex tutorial. Simply click the hyperlink and get inside. So, in this, you will be getting stepwise guidance to start the tutorial. So, see, these are the steps you can find in this Chromax tutorial in ligand receptor complex binding. So, if you see this, the first step, like however we have seen in molecular docking using Autodock, 6 steps we have seen. The seventh step I told you how to do the molecular docking analysis using Discovery Studio Visualizer. Okay, and here if you see in Chromax, we have 1, 2, 3, 4, 5, 6, and seventh is the analysis. So here also I have 6 steps, but we have so many sub steps inside. When compared to docking, okay, we have more steps here in MD simulation. So, but in MD simulation, we have problem like we can't able to see an output on a single day. Okay, so to execute this production molecular dynamic simulation, definitely it take minimum 24 hours. If your workstation is slow, then it takes even up to a week based on the size of protein. Some MD simulation takes 1 week to complete the protocol. So if you're working on this MD simulation, you should not do even browsing. You should not disturb the system. You should leave it as such. The server or workstation you work with the MD simulation should be dedicated only for MD simulation. You can't use it for any other purposes. And that too, I mentioned you better you should have a Linux operating system, and under the Linux operating system only, we have to work with Chromax using the commands. Okay, in the terminal, you can work with the commands. Now, let me take you to the step by step of ligand receptor, how to work with AMD simulation of ligand receptor complex. So, first thing what they are mentioning is they want to try with a sample file. The sample file, what they're mentioning is you can also download it from here. There is a hyperlink here. You can download from there. And in that, if you download this file, you will be getting, I can show you that. Okay, you can able to find, they've already cleaned this file also. Find from text demo, CADVD, clean. So just because you want to show me the coordinates of the file, I'm showing you now. So open with Notepad. So this is the best editor to visualize the coordinates. So whatever I downloaded, this is a coming under hydrolase protein, and two profile phenol. This is the ligand is already complex with T4 lysozyme. So our target protein, whatever I downloaded, is the T4, and the ligand, whatever we have, is the two profile phenol, whatever mentioned at the top. If you drag and see, okay, that you can able to find it from here. This is what HET-ATM, that propyl phenol. So this propyl phenol is represented by JZ4, and the amino acids of the protein, that lysozyme protein, is this. Okay, so as per gene is in position 163. So almost we have 163 amino acid protein. This is a 163 amino acid protein. The last amino acid is as per gene. Okay, and then if you see this ligand phenyl, that is represented by JZ4. Okay, so according to this tutorial, initially what they want is they want to split the JZ4 separately, okay, from this protein. So if you split the JZ4 separately from the PDB coordinate, then the JZ4.PDB is your ligand.PDB, and whatever you split from the ligand receptor, then the split thing, whatever you get is the target.pdb. So that is whatever initially you have is a protein ligand protein receptor complex coordinates, but after splitting that you can find ligand separately and protein separately. Okay, so actually you are using a command called grep, gradual. This is a filter command present in Linux operating system. We use grep command followed by JZ4. So it means that we are going to extract JZ4 coordinates from this PDB file, and we are going to output in the form of a new file name, or a new file name, whatever we create is JZ4. Initially, whatever I download is clean.pdb, but after creating and a file called JZ4, we are outputting it. So we are using this output filter, okay, to write our coordinates in this. So this is a command to write. Just like that, in Windows, what we can do, we can cut these coordinates, and we can open another notepad, paste it in another notepad, and we can save it like js4.pdb. Whatever I am going to we can do here in Windows, in Linux, we have to use this command, Linux command, and we have to follow this protocol. I told you here in MD simulation, if you can follow this commands step by step, however they mention carefully, we can able to get a final output in terms of RMSD, RMSF graphs, radius of gyration, all these things, whatever you're requesting. Here, according to this tutorial, we can able to see RMSD and RMSF. Okay. So what is RMSD, what is RMSF, we'll be discussing soon with examples. And we can also see the command like TAR. Okay, the TAR is used for unzipping the file. You know, we have WinRAR, WinZip. This is used for compressing the file or folder. If you want to uncompress, obviously in Linux we have a command called TAR. So we are going to unzip the kind of a file, and we are working on it. Okay, and we are using a Gromax command. We are using a particular. So if you want to work with a Gromax kind of a program, obviously one has to remember we have to start with GMX followed by what program we want to use, then in a kind of an parameter, and after the parameter whatever we give option parameters, this is an option parameter, then this is your file parameter, and then this is again an option parameter, then we can give a file parameter followed by another parameter. Like this we generally provide, okay, what to be given. This is actually parameter of the file, actually the content in the file. Okay, like this, whatever we want to work with the MD simulation, one should first have a knowledge on Linux operating system commands. If and if you know how to work with the commands of Linux, you can try with this MD simulation. And the next step is to go with the ligand topology. Okay, in the ligand topology, we have to practice this step. Okay, here in this step, we had hydrogens. You remember adding hydrogens we also found in Autodoc. In the initial steps, we add hydrogens to the ligand atom. Similarly, here also we do that. So most of the steps look similar to the Autodoc protocol, but finally we can see something variant with respect to Autodoc. Yeah. Any doubts? No. Okay. Now, similarly, In between we are using the Python programming, Perl programming. Here also we are using the Python and Perl programming in between. See the Perl programming. We are using a Perl command to work with the Perl programming. And even in that first step, whatever I showed, there also we have to download a Python programming file. If we have to change the coordinates, for example, if you are using a metal binding protein, we can't able to use the same protocol. We have to slightly change the protocol. That kind of a changing of protocol we can able to do only in the Python programming code, whatever you download. Here we also have a step to download the Python programming. So in the Python programming, if you know the Python programming, we can change the code with respect to the ion, and we can work even with ions in MD simulation, ion binding protein in MD simulation. Okay, some complexity is there, lot to know. At least first you try with the sample file if you have a configuration. Okay, if you have the hardware to work with it. Okay, so build the complex after adding the hydrogen, and then you have to follow the steps rigorously, and you do have to get the output exactly. If this output is not exact, if any one or two lines are missing, then there is some trouble in execution of your MD simulation. So you have to carefully work, and you have to check even the outputs however shown in this. In this tutorial, they have mentioned you to, they have guided stepwise, and also they have showed you what kind of output you can get. So this is an output. Okay, and similarly, if you execute it, what is the output you can get? Like this, input and output. They are mentioned in this tutorial. So after adding the hydrogen bonds, okay, the next is to go with the defining box. You remember I explained you on yesterday, like encapsulating your protein in a grid box. Similarly, here also you do that. The objective is to find the binding sites. If you are trying to dock the ligand with the receptor, if you want to check the XYZ coordinates of a particular binding site, not with respect to amino acid, but with respect to an atom of an amino acid, then obviously we should have enclosed this entire protein in grid box. And also to check whether it is hydrophobic, hydrophilic, distinguish the type of sites, one should have grid box, and this grid box is going to split in grid lines in major axis and minor axis. And now if you see in this kind of a different box and solve it, okay, we can use the Chromax command, and we can use the different programs, whatever I showed you in the table. And this is the format, okay. So to know more about this, even we can use that man gmx edit corner. You remember man? Man is the command used in Linux for manual. Like if you want to get help of a particular command, how to use the command, you can use man spacebar gmx, man spacebar gmx spacebar edit conf. This is how we try with this kind of a commands. And also you can use the Gromax documentation. The next step, the guided is it asks you to add the ions. So after hydrogen ions, hydrogen atoms, you are going with the solvation. Then only it's asking to add the ions. So after adding the ions, okay, using the steps, go with the energy minimization. In energy minimization, we use a protocol like charm, okay, and we can go with the energy minimization or whatever protocol mentioned. We also have Gromos. Gromos is also a kind of an energy minimization protocol. Use any kind of a energy minimization protocol available here, and then we can work with energy minimization. Later, we can go with equilibration. Under equilibration, whatever protein we keep, we have three important coordinates. So number of atoms of a protein is very important to know how a ligand is going to bind and how many sites your ligand is going to bind with the receptor. So number of atoms is important, and the pressure, we keep a constant pressure for all the proteins, whatever we work in MD simulation. So what is the pressure and what is the temperature? In chemical engineering, we have standard temperature and pressure. You remember about that? The temperature, we keep it as 298 Kelvin, pressure as 1 atmosphere or 1 bar, whatever it is. That is the default standard temperature pressure conditions, isn't it? Similarly, here also we use this kind of a standard conditions, and we do that kind of an equilibration. Okay, so whatever we work in wet lab, we are trying with simulation in computational. So we try to simulate in such a way that we consider all these parameters. So we also have a part of that is NVT, number of variables, okay, number of atoms, volume and temperature. V stands for volume, and earlier the NPT. After NVT, we have to go with the NPT. P stands for pressure. We keep a standard volume and temperature and the standard pressure and temperatures. Volume, you remember 22.4 liters. Like how are you used in calculation? Here you use a kind of a standard number of the these variables. And finally, you go with the production MD. In production MD, this is the final command we work, and was during this is similar to running the Autodoc. This is similar to running the Autodoc in doc. And then we have to go with the analysis. In the analysis, finally, we can able to study the output of RMSD, RMSF. Okay, so this is the command to get an RMSD image. The RMSD image is in the form of XVG. And finally, for RMSF, we can get like this. Okay. So now let me take you to the PPT and start explaining. Now at least I mentioned you how to find this information. RMSD. What is RMSD? RMSD of atom position is the measure of the average distance between the atoms. Usually, the backbone atoms of superimposed atoms. So this is what RMSD. And RMSD, to work with RMSD, this is the command we use, TMX RMS. And these are the parameters we use. So the RMSD formula is mentioned. Once in this graph, we can able to see that for a protein, whatever we worked, okay, so this is stable up to this. And our protein is stable up to this. If you draw a line, it should match the line. So if you see this, it comes around 1.1 something like that. So the protein is stable from this position to this position, nearly 160 to 200. And similarly, something around how much? Some 66 to 90. And what is the RMSD value for this? For this, it comes around 0.75. So the RMSD value is 0.75 for this time to this time, and RMSD value is 1.1 for this time to this time. And I tell you once again that RMSD is in y-axis and time in nanoseconds is in x-axis. Generally, if you want to show the MD simulation output, you have to work minimum of 100 nanoseconds. But at present, many of them are trying for 200 nanoseconds to 500 nanoseconds in good quality publications. Okay, to assess the output, we have to assess like this. First, we have to get the average RMSD value and check the time period. And this is another output published. Here, this is EGFR without ligand. What is that? It's shown in black color. So whatever black color line you can able to see, this is without ligand. But whatever the red we see, the ligand is bound with the protein. If only if the ligand receptor complex RMSD value is less than the protein alone, we can tell our ligand is stably binding. The binding is stable. If our ligand receptor complex is more, then you can tell our ligand is not binding stably with the receptor. This is the conclusion. Other than this conclusion, we can check the average RMSD value. What is the average RMSD and what is the time period? And what is the average RMSD? We have three different time periods. No, 1, 2, if this is stable. Okay. Similarly, I tried with a different protein of the same ligand. Here also you will be checking the red is coming below. So it is stable only, even for this receptor. But you find so many zigzag. When compared to this protein stability is good. That is the ligand bound with the receptor is good when compared to this. Okay, so the stability is less. See, so many zigzag. I can't get an average distance. And so we can go with an RMSF. This measures the deviation of the position of a particle with respect to the reference position. Actually, it tells about the carbon alpha fluctuations. RMSF stand for the carbon. I need just 3 to 4 minutes to complete. Carbon alpha fluctuations. Here you see this is the most observed fluctuation. That too, whatever observed fluctuation is around 0.55. If your fluctuation, C alpha fluctuation is above 1, then how many fluctuations you find, you can tell that much of C alpha fluctuations. So actually the C alpha fluctuation is now at present from 94 to some around 1 to 110, 115. So 94 to 115, it is unstable, but it is less than 1, so there is no problem. We can ignore this and we can tell our protein is stable with respect to C alpha fluctuations. And here, other than just simply working with RMSF with a protein, here I tried with ligand also. That is, if a ligand receptor complex and protein alone. So if you see this, we started with this, but later we can able to see less fluctuation only. We didn't find more than one, but which is having more fluctuation. This is less when compared to this. The protein alone is showing more fluctuation. So you can tell the C alpha fluctuations is negligible here in this case. But see, in the second case, we have that protein, ligand protein is touching more than one. So C alpha fluctuation is more. This is not actually preferred. And radius of gyration tells about the axis of rotation, a radial distance. So it actually measures the moment of inertia. This is going to measure the moment of inertia. Okay, and this is the formula, and we can able to check the average radius of gyration, and we can report it. And this is the command we can use. So this is the fourth output, binding free energy determination. In the binding free energy determination, we can able to find the binding energy even in MD simulation in the form of one trajectory, three trajectory, two trajectory. So in one trajectory, we have only one file, that is ligand receptor complex. We have a ligand receptor complex in one trajectory. But in three trajectories, we have ligand receptor, we have a this is receptor, ligand receptor, receptor alone, and we have ligand alone. We should have three different files, one file, second file, third file. If only we have three files, for all the three files, we have three different energies. If you have three different energies, we can able to use these three different energies and calculate the del G binding. In three trajectories, we can able to calculate del G binding as the del G complex minus del G receptor minus del G ligand. So this is applicable for three trajectories. This we can able to find in MMGBSA. Then what is MMPBSA? What is MMGBSA? That is mentioned here. See, MMGBSH molecular mechanics generalized bond surface area. MMPBSA molecular mechanics Poisson-Boltzmann surface area. This is linear interaction energy. That's what they mentioned here as LIE. So this is actually used for two trajectory approach, where you have a receptor, ligand, and the ligand alone. If you use these energies and you calculate the energy, that's what LIE. So these are some of my outputs of my project. I tried with multiple ligands, okay, and showing the kind of a result. The one thing which I missed is principal component analysis. If I found more RMSF fluctuations, okay, and I found many peaks above one, then I suggest you to go with principal component analysis. So in this principal component analysis, APO stands for the protein alone. If this is for protein alone, which is showing in green, and if much of the green is not masked by the ligands, then you can tell this is the reason why we have more fluctuations. So principal component analysis we can use if you have more fluctuations. Okay, I've completed my session. So if you have any doubts, you can ask me. Any doubts? But okay, thank you for your patience, okay, and interaction from the beginning. So tomorrow we'll be discussing a new chapter, that is on vaccine design. Today I have completed drug design and molecular mechanics in detail. Okay, any doubts, any accumulated doubts, you can also ask me in WhatsApp so that I can mention you in the forthcoming session. Thank you. Have a great day to you all.